In this lesson, we're going to learn about action potential of the cell membrane, and we're going to discuss it directly on this coordinate plane. The vertical line stands for the membrane potential, which is the difference in the electrical charge between the interior and exterior of the cell. And the horizontal line stands for the time, which is measured in milliseconds, along with the membrane potential, which is measured in millivolts. So the events that are occurring in this process are on the cellular level, and they are measured in very small units. The charge of a membrane at rest is negative 70 millivolts, and for an action potential to occur, this number should rise to negative 55 millivolts. So negative 55 millivolts is the minimum requirement for an action potential to start. It's called the threshold voltage. So the first peak in action potential is rising the voltage of the membrane from negative 70 millivolts to negative 55 millivolts. And then at this point, the first step of the action potential is going to start, which is the depolarization step. In the depolarization step, the charge of the membrane goes toward positivity. And this step of the process starts when the sodium channels start to open, and they're going to enter the cell. They usually have a higher concentration outside the cell, so when the sodium channels open, they start entering the cell and bringing the cell toward positivity and far from the membrane's resting potential. The entering of the sodium ions to the cell creates a positive feedback loop to open more sodium channels, and this further depolarizes the membrane until the membrane reaches about 40 millivolts. In a mature neuron, the depolarization step lasts about 1 millisecond. After that time, the sodium channels start to close and they become inactivated, and they're no longer able to flux the ions inside the cell. And just as they get inactivated, the second step of the action potential starts. It's called the repolarization step. When the sodium channels get inactivated, the potassium channels start to open. Potassium ions are usually higher concentrated inside the cell, so when these channels open, they start going to outside of the cell, leading the cell toward a negative charge or toward the resting membrane potential. There is only one thing about the potassium channels, and it's that they have a much slower kinetics comparing to the sodium channels. So after the sodium channels get inactivated, it takes about 1 millisecond for the potassium channels to get opened and start migrating out of the cell. This process of migrating potassium ions outside to the cell continues until a signal has been sent to the potassium ions when they get near the resting membrane potential. As we know that the potassium channels have low kinetics, so when they receive the signal, they take more time than required to stop the repolarization step. This makes the cell to enter the third step, the hyperpolarization. This step brings the cell toward more negative charges than required, and the cell enters to a slightly more negative charge than it's required for the resting membrane potential. Now the cell should enter its resting membrane potential, and how it can go from the hyperpolarization state to the resting membrane potential. This usually occurs by 3 natural steps of the cycle. The first one is when the potassium channels get totally closed. The second one is the natural permeability of the cell. That means the sodium and potassium pump. This pump constantly exchanges these two ions inside and outside the cell, according to the cell's requirement. And usually, the potassium ions inside the cell are higher, and sodium is higher in the extracellular environment. The third one is the concentration gradient, in which is the movement of particles from higher concentration to lower concentration. These all work together to bring the cell to the resting membrane potential, so it can be ready for another action potential.